And this was when we were getting at the idea of there's some misconceptions on, previous data, saying, oh no, the uterus is sterile. And so other scientists said, but wait, no, it doesn't look like it's sterile. And then other ones said, oh no, you're wrong. So there's still some debate about this. But I just inserted after this slide, there's another paper that was published, and this one is just from last year. And I thought it was interesting because it talks about the extracellular vesicles that we talked about back in quorum sensing and nanotubes and extracellular vesicles. And so here they're saying that the microbes of the mother are excreting vesicles that can make it into the uterine environment, into the amniotic fluid. And so they suspect that this is, the infant will ingest that, and that it's a way to have some, they suspect it's helping the fetus, their immune system, kind of getting it primed and working a little bit with these extracellular vesicles. So I thought that was an interesting paper that was just published. This is kind of the synopsis of that paper. The mechanism between the maternal microbiota and the fetus, potentially playing a pivotal role in priming the immune system before birth. So there will be more to come, I think, on this topic of what's going on in the amniotic fluid and the fetal environment. But that was a nice study that was done, nicely done. Okay, so zooming forward now into, where did we go? Here. So now further down into the slide set, I think this is where we left off, where we're looking at, we made our way through the mouth, and now we're getting into the gastrointestinal tract. And so this is probably the part of the human body that's received the most effort in terms of grant funding and research, looking at what's going on in the gastrointestinal tract. And so let's start kind of at the top. After the mouth, you get into the stomach. And I think we all appreciate, if you've had some basic physiology, that there's high acidity in the stomach, down to, was it pH 2 to 3 or something like that. And so because of that, primarily because of that high or really low acidity, I should say, or low pH, there are not many microbes that have adapted to that. So Helicobacter pylori is the most famous example. It's not the only one, but this is the one that has received the most attention. That one can survive at pH 1, which is quite acidic. And it's partly doing that because it can kind of bury into the mucus lining our epithelial cells in the stomach. And so it can kind of cozy into the mucus and get covered by that. And it can also excrete certain chemicals that can raise the pH right around the cell. And so it can kind of create this little microenvironment of a slightly higher pH. So this one, its claim to fame is that it can get ulcers, potentially. Now, just because you may have this in your stomach doesn't mean you'll get ulcers. There's not a direct link, but they do see that this one can cause ulcers. And this was very controversial when they proposed this idea, because the thought at that time was that you get ulcers from eating like spicy food or stress, and somehow that messes with your physiology. And then they actually, it was, I think, what's his name, Barry Marshall, it's this guy. He, Barry Marshall. So he actually used himself as a guinea pig, and he swallowed a vial of Helicobacter pylori. He grew it up and swallowed it. And he said, I'll prove to you that Helicobacter causes ulcers. I think they did this on stage at a conference, actually, which is like way too into their science. They're willing to swallow a bunch of Helicobacter. And he proved he got ulcers. Now, luckily, he did this back, and I think it's still the case. Helicobacter is still sensitive to antibiotics, but luckily, that was the case and took antibiotics and was able to get rid of it. But nothing that I would propose to do. I mean, I love science, I love my research, but I would not go that far. So this was a huge turnaround looking at the idea that this microbe can do that. So this microbe has received a lot of attention for that, and then it got this evil, oh, it's evil. We have to destroy Helicobacter. It causes ulcers. This is awful. And so there's been this camp of scientists who think that that's the way we should handle Helicobacter. But more recent thoughts on this idea is that we now know that humans have had a relationship with Helicobacter for thousands upon thousands of years. And that if Helicobacter has been hanging out with us for that long, there's probably some good reason for that, and that we shouldn't be so quick to kill it off. And so scientists are starting to think more from an evolutionary perspective and an ecological perspective on the microbes that coexist with us, and trying to get out of this knee jerk, we have to kill it because it might cause ulcers in this case. So if you are doing certain things that can raise your acidity, or I should say raise the pH, that raise the pH of your stomach, you have to be a little cautious about that, because it can happen if you're malnourished. That can happen just naturally if you're malnourished. And then if you take too many antacids, it can also cause the pH to rise closer to neutral. And that's not good. You want an acidic environment. And so if you think about some of the pathogens that you get exposed to, or potentially can get exposed to, like Vibrio cholerae, that one could easily then survive to pH that's not so acidic, and then make it into your intestines, and it can make you really sick and can even kill you. So people who pop antacids like it's candy, I don't think they understand that this can be a risk, a big risk. And so that's something, if you have someone that you know or a family member that's routinely eating those, they should be cautious about that. So I put in a couple slides here with the discovery of what really causes ulcers if you're curious. It's just more for your entertainment. So, okay. So the stomach has limited colonization. Now, if you get into the intestines, that's a different story. And so you start with the small intestine, and then you work your way to the large intestine, and then to the cecum or colon area, I should say. So there's different types of microbes, different populations of microbes in these different areas. And so with the small intestine, well, in general, in the entire intestine, some people will say up to 10 to the 13th. So 1 times 10 to the 13th bacteria per gram of feces, per gram. That's a lot. And so they typically throw out this data, about 50% of feces by weight and volume is composed of bacteria. And overall, there's more anaerobes by far, that type of microbe. And so it's kind of weird, because your intestinal tract is open. It's an open area in the sense of it's a lumen essentially, but there's so many areas of the intestine that can be anaerobic. It's not that hard, especially when you think about the size of microbes being so tiny. And so what tends to happen is the oxygen that is diffusing from our cells that make up the lining of the intestine, the epithelial cells, usually the facultative anaerobes scarf that up really quickly, because of course they want oxygen. If you can use oxygen, you use it, because it gives you the most ATP. So E. coli, if it's smart, will scarf up all that oxygen, and then locally there'll be this drop in oxygen, and then the anaerobes come in and survive quite nicely. And so typically we do see E. coli colonizing quite rapidly so once a baby is born, very quickly E. coli starts to get established, and it's not very long before you have, because it's facultative, it will preferentially use oxygen if it can, but then it can persist once oxygen is used up. Plus, now that oxygen is used up, it allows the anaerobes to get in and set up camp. So bifidobacterium is an anaerobe, Bacteroides, Clostridium, all of these can be found in the gastrointestinal tract of humans. And most recent studies are saying about 1,500 different bacterial species, but I think that number will go up. More careful studies. And they say we do have methanogens that make methane gas. We do have that. So those are archaea. Those are not domain bacteria. And so we do have a mixture. We have some fungi as well, and lots of viruses that hang out in the gastrointestinal tract. So this is just a mock-up of the gastrointestinal tract, and this is trying to get across the idea that you find different environments depending on what part of the tract you're looking at. And so you can see the stomach has very low pH, pH 2, but then when you shift into the small intestine, you see an increase in the pH. And then the large intestine is closer to neutral. And so I don't care that you know all the individual parts. You don't need to know all this, but I do want you to know which connects to what. The stomach connects to the small intestine, which connects to the large intestine. Okay. The other idea here, is that you can see this shift of different microbes. And the person who made this figure, they were trying to really make the point that it's in the large intestine where you see great diversity and very high populations compared to any of the rest of the tract. And so the large intestine wins in terms of sheer numbers and diversity with the number of different species. So it's a little challenging to try to study the different parts of the gastrointestinal tract, like who lives where, how many, what's the ratio, because primarily what they do is they sample feces. And so that's really hard to tell where did the bacteria in the feces come from exactly. So that's been a challenge unless you work with organisms that will get euthanized, and then you can do surgery on them and actually figure out. And so with mice and rat studies, that's doable, where they will euthanize and actually take tissue of the small intestine or the large intestine, and they can more precisely say who's living where in that environment. One of Dr. Lafferty's former students, he was able to do that with bears after they were hunted and killed by the hunters. Some of the hunters participated in a study where they gave permission for her to take samples of the small intestine and large intestine. And so that was another way that she was able to get good data. But with us, can't really do that, right? So how do you figure out what's going on in the human? So far, it's primarily fecal studies that we're doing these analyses. So you got to keep that in mind because that's not super precise. This is another image, and just giving a little bit different perspective of the gastrointestinal. So it's been a while since you've looked at anatomy of the human. I just wanted to put that in there. But again, they're showing correctly that the stomach has the fewest amount of microbes, 1 times 10 to the 1, 10 to the 3 per mil. Then you see it jump in the small intestine to 10 to the 3 to 10 to the 7. And then by the time you get to the large intestine, it's what, 10 to the 12 to 10 to the 13 per gram. Per gram. That's a ton. Okay. So also to think about is a healthy gastrointestinal tract will have a mucus lining. And so all of the pretty colors on the left side of the image, those are microbes. And then you see the green stripe, that's the mucus. And then the blue is the intestinal stuff. And you can see some microbes are in the mucus. The red popping against the green here, blue. And so they are able to get into the mucus to some extent. That's possible. But you can see that, most of them are coming up to the mucus, but not penetrating into it very, very much at all. Some microbes are very good at eating the mucus. That's part of their nutrition. They like that as a carbon and energy source, and that can be a good thing. That's fine. That's not a problem. If things get out of whack, out of balance, then it gets a little dicey. So again, remember, there's a lot of surface area when you look at the intestinal tract. The villi have incredible surface area for all kinds of places for microbes to hang out and live their life. This is a nice image that was taken for a small world photography contest. That was really nice example. So they're kind of wondering, like, why do the microbes keep a polite distance, so to speak, from the intestinal cells, our epithelial cells? And they talk about force field. That's what they're calling it right now. And so they think they're carbohydrate binding proteins somehow involved in this. That's kind of keeping the microbes at bay and making sure that they're not encroaching too deeply. So that is still very tentative. They need to map that out a little bit better. But the fact that microbes are kind of keeping this polite distance, they're not trying to get too close. So this is just, again, trying to point out the idea that you have, from a microbial perspective, we're talking, really small distances between the microbe and the epithelial cells that are, your eukaryotic cells that make up the intestinal tract. So there's certainly potential for the microbes that are right there in that mucus lining or right next to it. They certainly have the potential to influence your epithelial cells. And we'll talk a little bit more about that. Also, there's been a lot of debate over the years about the appendix. Some people think, oh, it's just a, leftover thing and doesn't really matter. Some people might have had, the appendix taken out or had your appendix burst. That happened to my brother a couple of years ago. And he had to do, undergo emergency surgery. And they actually think now that it's good to have an appendix and that if you can keep it healthy and intact and not have it burst, maybe that's a good thing. And so they think maybe it could be like a reservoir for holding microbes that might need to be used to replenish. Maybe something that happened with your intestinal tract where some of your microbes crashed for one reason or another. Maybe they could help repopulate your intestinal tract. So that's the current hypothesis that's gaining acceptance. So the techniques that they use and the technology that they have to study all of this, they are finding, especially when they do studies in mice and rats, they are finding that some microbes can have some pretty potent or powerful impact on the host. And so I just wanted to point out a couple of studies. There's one that was done with Akkermansia muciniphila. This one is gaining a lot of scrutiny. It's gram negative and it's an anaerobe. And now they're showing that just this one microbe has a pretty profound influence on making sure that the intestinal tract is growing and maturing the way it should. And so this, again, they're doing this in mouse and rat studies where they can show presence of this one microbe versus absence of it and what could happen. And so what they do is a lot of this research in mice and rats, they create germ-free, they actually call them germ-free mice, where they're housed in a totally sterile environment, like, in a bubble of sterility, which has to be extremely demanding to care for those animals. Everyone going into those rooms has to be fully garbed up, and you cannot be transferring any microbes from you to the mouse colony. It's got to be quite intense. But anyways, so they have these germ-free mice, and then they will, in this case, they gave Akkermansia to these mice to see what would happen. Usually they make the mouse swallow it, and then they wait to see what happens. So in this case, they're showing this one has a lot of impact on the gastrointestinal tract and how it matures. Here's another one, Bacteroides theta iota omicron. which has, again, received a lot of attention. And this one they know very good at breaking down a lot of carbohydrates that you don't have the enzymes for. Your eukaryotic cells don't have the enzymes for. And then some of that breakdown product is good for you because it's part of your nutrition. So they use it for some of their nutrition, but you can also use the breakdown products. So there are these types of studies where they're looking at what happens with this particular microbe. And through a lot of these studies, they quickly realized when examining the data that, wow, they really can influence the gastrointestinal tract, the maturation of it, the immune system. A lot of our immune system is in our gastrointestinal tract. It's a phenomenal amount of our immune system is found there. And they're finding that these key microbes can really play an important and impactful role. Well, since some of these studies, there's been a lot of other studies where, again, they just manipulate one microbe into these germ-free mice. And so these are just snippets of published research papers. And you can see most of these are talking about just one microbe and the impact that one microbe. And so, again, these types of studies are doing a lot of this because they're trying to figure out, what does one microbe do overall versus the consortium? And are certain microbes more important than others that are in gut? This one in particular, Lactobacillus reuteri, has received a lot of attention. They have been looking at that. It seems like it's able to have a very strong positive impact on their human host. And so if you ever take probiotics, you might want to look to see if that, so far I've only found one probiotic brand that has that. So, okay, so they've been using these, one species at a time. What happens when we put this one species in to a germ-free mouse? So don't forget, when you start thinking about all the microbes that are in us, we're talking about the gastrointestinal tract, and that's where they need to stay. And so you can have issues where if you get into an accident, like a car accident or something like that, where you get injured and some of the microbes from your gastrointestinal tract get into other areas, then of course that can be an opportunistic situation where it's in a new part of the body. That part of your body and those microbes haven't co-evolved, and so things can get a little out of hand. This is a case of gas gangrene starting to happen on the abdomen area of a patient. And so there's this idea that the microbes that are in our gastrointestinal tract, most of them that are there are actually doing very good things for us as long as they stay in the gastrointestinal tract. And so they've looked at this one too, because they're realizing Bacteroides, some of the earlier papers are like, oh, it's really good, it seems to be very beneficial. And then other papers, the data were showing maybe not. And so they started to look at this genus a little bit more carefully. And basically, Bacteroides fragilis. They said it's the same as with real estate: location, location, location. And so where it's found is really what's going to determine if it's good or bad for the host. And so one microbe in the right spot can be good. Get it in the wrong spot, it can be bad. And so I just want to point out that you can't always say this one microbe is always good or always bad. You have to put it into the context of where it's located, what's it doing, does it normally belong there or not. And so these slides, I'm just trying to point out that in the right spot of the body, it can be considered a commensal, it's helpful. In the wrong spot, it acts as a pathogen and can be very virulent. And then the nitty-gritty idea, you're wondering what that meant. They were just talking about the Bacteroides, those species. There's been a lot of issues with the taxonomy. It's just been a headache trying to get them all mapped out in the phylogenetic tree. So that's what they meant by the nitty-gritty. But that's not really relevant to what I'm trying to get across here. So basically, I like this quote from the paper: with the adaptability of a chameleon, the Bacteroides species can function as an integral partner in the human metabolic system, and yet can be the cause of serious life-threatening disease. And so I think it's important to keep this in mind when you get into this kind of research or read about it. No one microbe is wholly good or wholly bad. You have to look at the situation and where is it located. So keep that in mind. Here's another idea of when things go wrong. They're finding even with just emotional stress, you can cause shifts in the microbes in your intestinal tract. Certainly your diet can cause a shift, and certainly antibiotic therapy can cause a shift in who's there and how many are there. And so the word that's often brought up when you start to mess with the populations and the diversity, they start to use the word dysbiosis. And I do want you to know that. Dysbiosis. So that just means an imbalance. Imbalance. Here they're showing pseudomembranous enterocolitis. That's not what your intestinal tract should look like. It should not be all bumpy like that. This is severe. Poor person. It must be miserable. So this is caused by C. diff. You might have heard that nickname. It used to be Clostridium difficile. Now it's been reclassified as Clostridioides difficile, but it's still C. diff. It's C. diff. So you might hear, especially people working in the hospital environment, C. diff is a problem. And it's gotten to be a big problem in some hospitals. Like they search for it. That's one of the things they look for when they grade hospitals. Like if they're at the A level, B, C, or failing level, they will look for C. diff and see how prevalent it is, and it can be a problem. So my sister-in-law is a nurse down in Green Bay, and she has many stories as we get together over the summer during vacation. We have a lot of stories about patients with C. diff and how hard that can be. And now they have to implement really strict protocols on how to like clean the room. That patient, once hopefully they get better and release, what they have to do to that room, because they've even found C. diff like on the walls of the patient's room hanging out. And so it can just like persist and hang out and be viable for a long time. So if there are these cases of biosis and imbalances, something's going wrong. So there's been a lot of thought about this probiotic. Maybe probiotics would be a way to help that patient get back to normal with the types and amounts of microbes. I think most of you have heard the word probiotic by now. And so, living microbes. And so the idea here is very often they're sold as pills, but you can eat probiotics in fermented foods. So that's another reason why I wanted you to have the option of doing that fermentation extra credit, because I wanted you to think about fermentation as a part of your diet, because I think that can go a long way to keeping you healthy. Certainly getting it through fermented foods. Probiotics are also sold as supplements, and some are better than others. We'll talk more about that. But very often, at least some of the earlier probiotics that were sold early on, most of them contain Bifidobacterium and Lactobacillus species, which is fair. I mean, those are common microbes. Many different species within those two genera hang out with you on a day-to-day basis. And so Bifidobacterium's on the left and Lactobacillus on the right. So fermented foods can have these as well: yogurt, kimchi, and kombucha. There's just a published study on kombucha. Oh, they were linking regular intake of kombucha and weight loss. So I don't know. They said it's early days yet, but some of the early studies were promising. So we'll talk more about fermented foods. So Or even looking at probiotics as a way to manage depression and anxiety. And so that has really captured the attention of some scientists that are working in this field of study, and psychologists. We'll talk more about depression and anxiety in a little bit with regard to the microbes that hang out with you. So there is this idea too, where some scientists are saying, you know, slow down a little bit. You know, how much do we really know about these probiotics? Is it really safe to be ingesting these? And so that's something you need to look into. I have my own personal opinion on it, but this article, I read through it. I mean, it's valid what they're saying, but I'm not sure I agree with everything in it. But that's something that you should read. My own personal philosophy, I do take probiotics on a regular basis, and I try to eat a lot of fermented foods. But if you have certain health issues, maybe not. Maybe this isn't. You know, I don't have any serious health issues I'm struggling with, so I feel like it's a fairly safe bet. But your doctor may say otherwise if you're dealing with some other issue. So again, a lot of this is this data is coming from mouse or rat studies where you have germ-free mice, and then you often compare them to conventional, they say conventionally raised mice, which are just mice that they just have the normal colony housing, and they get their normal microbes acquired from their mom and their litter mates, and people handling them, I guess. So I just wanted to make sure they often use the term nanobiotic, also known as nanobiotic. So sometimes they'll throw that jargon in when you read a paper. So germ-free is just easier to say. So here, this particular paper, they were looking at what does it do if you have an absence of microbes versus a presence, and they were zooming into the immune system. They're saying, you know, germ-free animals have some serious problems with their immune system, and then they get into some of that. So a lot of what they're talking about in that first few sentences is all immune system cells. And then they're saying if you take those germ-free mice and you start to dose them with gut microbes, you get a restoration. So it's not like they're done, they're permanently going to be at trouble with their immune system. They're saying you can restore it. Put the microbes in and they bounce back from it. So that's nice. And so most of this, I'm not asking you to know all the immunology terms here. It's the big picture idea. And then that last paragraph, colonization of germ-free mice with a single species, Bacteroides theta iota omicron, can restore the mucosal barrier function. So again, there seems to be certain microbes that are like Superman. You know, they're just really potent and powerful at influencing the host in a very positive way. So the idea here of the early work with looking at gut microbes, some of the scientists were looking at, is there any link between what's going on with the gut microbes and obesity, which is essentially a pandemic, really, when you think about this issue. And it's not easy because they do think that obesity is the result of many, many things, all playing a role that leads to obesity. And the microbial composition is just one part of that. So Jeffrey Gordon did a landmark study where when it was published, it really caused a firestorm of excitement with regard to, wow, this is really a profound impact on the idea of obesity versus leanness. So this was done in mice, so that there's that limitation. But basically, this is some of the results from his study. And this is many years ago when the technology, it was expensive. So he only took it to phylum and essentially to phylum level. He didn't go all the way down to species level. But you can see just by comparing these two pie charts and looking at those two cute little mice, there's an obvious difference here in terms of the ratio of the microbes. And so with the obese, you see a lot of the Firmicutes species and far less Bacteroides phylum. And with the lean, it's pretty much the opposite. And so that was the landmark study where it really captured the attention of other scientists. Like, wow, that's a big difference. And of course, the actual obesity versus leanness was very, very noticeable in the cute little mice there. So then they said, okay, mice, sure, we do a lot of work in mice. What about humans? Can we do it in humans? And so the best way to do this is using identical twins. If you can do that, then you control for a lot of other variables, not all, but a lot. And so monozygotic got identical twins. And what they did was they took fecal samples from the twins and they transplanted them into germ-free mice. And then they followed the experiment out to see what would happen. And so with the obese twin donating fecal samples to germ-free mice, the mouse ended up obese. With the lean twin, the lean fecal sample into the germ-free mice, they stayed lean. So again, that was a pretty profound study where they were showing you can transfer obesity from the microbial composition found in the species in this particular case. So when they started looking a little bit more carefully and doing further experiments with identical twins, so this first portion on the left here is what I just described. Then they decided to do some studies, further studies with those mice. And they said, what if you do that fecal transplant and then you co-house those mice, the mice that got the obese species fecal samples or the lean, what would happen? And then they said, what would happen if then obese, lean, and germ-free all living? What would happen? And so with the obese and the lean, so this middle portion of the bar graph, look at the difference. The green bar graph, this is the obese, there's the fat graph. When they're co-housed, less fat, the obese mouse got way less than if it was just housed on its own in a separate cage. And so that's a huge significance. So what that's implying is that the lean mouse, maybe its microbiome, somehow had a stronger impact and could keep. You remember now, mice eat each other's species, they groom each other, so there's transfer going on. And so they're sharing their microbiome. And the fact that the obese mouse didn't gain as much fat, they're saying that the lean mouse, its microbiome, probably had a pretty strong impact and was able to kind of regulate and modify how much fat that mouse was going to get. And then that last lost weight here. And so again, this germ-free mouse, it actually lost weight when it got put into the mix. And so again, they're showing this idea that the feces from the lean twins seem to have a strong impact, and in this case, a positive impact in creating less body fat when they're co-housed. The other thing too that is something I want you to really key in on is that the obese human microbiome was less diverse. And I'm going to talk more about that as we get into this in more detail. But that less diverse microbiome, over and over, studies are showing that is causing problems for the host. And I'll get more into that. The other thing they were able to demonstrate is that the microbiome, the microbes, they could, the fancy way to say it is they increase the capacity to absorb energy, which just means that those microbes were very good at taking the food that the host was eating and creating lots of caloric products that the host could then take advantage of. And so basically those microbes were really good and efficient at using the food that the host was eating and was able to extract a lot of energy for that, which it shared with the host, which meant the host had all this extra energy and started to become obese. And so there's this idea that some microbes are just really efficient in their physiology and can really extract a lot of energy from foods that you eat. So then the bar graph is showing that when you co-house the obese with the lean, they gained less weight. And so they said, and then when they actually analyzed the microbiome, it looked like the lean microbiome had taken over and was dominating. And then the germ-free mice actually lost weight. And so they said, you know what? We think that the lean microbiome actually had a greater fitness. They were just had a more powerful impact on the whole, all three, the obese and the germ-free. So this was, again, this study was done fairly early, but a very elegant study, not terribly complicated in how they ran it. And they were able to get some really good data. And so when this hit the news, it made a big splash and people started to really think more carefully about the microbiome and what it could be doing, which then it kicked off all kinds of studies that were being done. Here's one that was just published last year, again, looking at diversity of the populations in your gastrointestinal tract in particular. But here they're looking at what does low diversity versus high diversity do if you're challenged with a pathogen. And so here they're just drawing to try and get at the kind of summarizing their study. And they're proposing that when you have here on the left is the low diversity, many different colors. Here you have more diversity of the microbes, and then the red rod shape is your pathogen. And so what they're saying is that when you have higher diversity, what they're also finding here, they're looking at the nutrient profile of the pathogen, the different colors, and they're saying if you have a lot of high diversity, then those microbes are probably going to eat, or they're going to get all these nutrients if you have lots of diversity. So then the pathogen can't get access to those nutrients as easily because the other microbes are scarfing it all up. And so it's a kind of you're out competing the pathogen essentially. You're out competing them because you're able to, with the high diversity, they're scarfing up all the good food the pathogen wants and needs. And so if you deny that pathogen its food, it won't persist and your immune system will have a better time killing it off. So this high diversity idea, this is just, looking at, if you get infected with a pathogen, what it could do for you. So we'll come back to that high diversity idea. So this was another study that was really early on. Or no, this is the same one, 2006. And so here they're looking at that idea of Firmicutes and the Bacteroidetes, looking at the ratio of those two, and under the obese versus the lean. Our results indicate that the obese microbiome has an increased capacity to harvest energy. It's really good at extracting energy from the food and giving it to the host. Furthermore, this trait is transmissible. Colonization of germ-free mice with an obese microbiota results in significantly greater increase in total body fat. And so it's transmissible, which was also a huge surprise. That was not something that they thought was going to happen. So this idea has led to some scientists saying, you know what we eat is part of it, but you also have to think about what are the microbes doing with what you eat. And depending on the microbes in you, some microbes aren't so good at extracting energy from the food you eat, which should lead to a more lean state. But if they're really good at extracting energy from what you eat, then they have extra energy to get back to you and could lead to weight gain. And so they're really starting to say it's just not calories in. You can't do that anymore as a way to think about whether or not you're going to eat. You have to think more carefully about which bacteria are hanging out with you, how many are they, and what's their physiology? What are they doing with the food that you eat? So the research on this is still early, but I think they're going to quickly find some very good results that can be implemented to help regulate our weight a little bit easier than all those diet schemes that are out there that never seem to really work. So, all right, we'll stop there and get into that a little bit more on Wednesday.